All right, so welcome to DevoWorm for this week. We haven't had a regular meeting in a few weeks, but today I'm going to talk about the Artificial Life 2025 conference. I'm going to go over some of the highlights and just kind of talk about what they did this year and maybe how that might benefit DevoWorm. I'm also going to revisit some topics that we've talked about in meetings past. I've been going through different meetings, our archives, and I've been looking for common themes. That's what I often do, is try to discover themes so we can revisit them, perhaps produce papers or talks or whatever. So that's what we'll do today. So this is the website for Alife 2025. The theme was Ciphers of Life. So this was held in Kyoto, Japan, plus online. So some of the talks were online, but most of them were in person in Kyoto. There's a proceedings. I didn't want to go through it because it's like 200 pages, but the proceedings are available for free on the website. So these are the proceedings. It's 879 pages, and kind of goes through all of the organizers and program committee and all this. And then, there are all sorts of papers on cellular automata, robotics, genetic algorithms, different types of artificial life platforms. Also, there's embodiment, all sorts of things. And so this is great diversity of papers available. So if we look at the program, there were several keynote speakers. There was Hector Zenil, who does algorithmic information theory, and he's talking about the title of his talk was Common Misconceptions of Computability and Algorithmic Complexity, a new old framework for causality, living systems, and scientific explanation. There is Blaise Guerra de Arcos, who does artificial life and artificial intelligence, and are they really artificial? There is also the Artificial Life and Art panel by Dr. Ikegami. They usually have an art feature every conference. Sometimes it's like a keynote. This time it's a panel. And so, they're talking about different ways you can make artistic media with Alife, with evolutionary algorithms, with different types of adaptive programs, and that sort of thing. So in this case, they're talking about the lineage of Alife and generative sound. So this is, it's a tool that's been used for a wide range of artistic pursuits. This is the Artificial Life Institute panel, Zen Philosophy, Plurality, and Artificial Life, and their roles in organic alignment of AI. So this is where they're trying to apply the philosophies in Zen Buddhism and plurality to build a roadmap to coexistence with AI rather than domination or hierarchical control. And then, is it possible to evaluate alignment with these philosophies in AI safety research? So this is what they're talking about here. So all sorts of different workshops, mortal agents in Alife, physical, psychological, and social death in the machine. This is social and environmental good, societal outreach, goal-directed behavior in living and non-living systems, emergence and evolution of language and communication, matter-information interplays, goal-directedness and metasystem transitions. This is Tonal 2025, which is thriving on noise and artificial life. So this was all about noise and its role in cognitive science and artificial life. There is Chemo Life Forms, which is the 5th workshop on chemistry and artificial life forms. Making Time 2, Temporality in Natural and Artificial Systems. Planet X, or the Cultural Evolution of Planet X. Emerging Creativity and the Wisdom of Crowds. Performative AI, Human-Machine Agency. Generative Artificial Life, Simulation of Real-World Computation. The ABM Hub Workshop, which is on agent-based modeling. SBAI-10, which is self-organization, and nano and electromechanical robots towards the robosphere. And then Alife and organizations, navigating societal transitions through co-creation. This one is interesting. I don't know if they have a description of it beyond actually there's the workshop website. So this is basically about sort of this open-ended cultural evolution and sort of the what ifs and possibilities of cultural evolution, perhaps on other planets. We are interested in the relationship between biological and artificial cultures. Can the cultural models evolved in robot swarms produce a plausible model for animal systems? How can insights from how culture evolved in animal systems then in turn help us harness cultural evolution as a tool for learning and creativity in robot swarms? So their interest is, they're just kind of focusing on robot swarms, but there are all sorts of different ways that we can think about cultural evolution, including vocal learning, nest building, tool manufacture and use, foraging behavior, and of course, movement. And there are all sorts of different aspects of culture that they'll focus on. It's like transmission biases, cognitive transformations, social structures, and processes. If you go to the workshop website, there's a big focus on open-ended cultural evolution, and in this case, developmental complexity in cultural environments. This is another interesting one, tonal, or thriving on noise and artificial life. Their interest is in exploring randomness in artificial life. Our central conjecture is that the role of randomness in noise is vital and irreplaceable. From stochastic mutations that fuel natural and artificial evolution to chaotic dynamics in numerical models, inherent unpredictability often lies at the heart of emergent complexity. So this is where just considering the different sources of randomness in artificial systems, in living systems. And so, thinking about the mechanisms by which random variation generates novelty and creativity, strategies for maintaining robustness amidst chaos, the flexibility achieved by the presence of beneficial variable noise and perturbations, sources of intrinsic randomness in natural and artificial systems, the history and present of the edge of chaos and self-organized criticality debate. That's an interesting kind of historical debate about how self-organization proceeds and how, if things can exist on the edge of chaos, that is, quasi-ordered systems that can also exhibit chaotic features. Then there's the role of noise in enabling greater integration and alignment and functioning across scales. And finally, the techniques for introducing controlled stochasticity to achieve open-ended evolution. And this, of course, is the program. So, there are a number of talks on neural noise, creativity and learning-driven self-organization, noise and multi-scale alignment, indeterminism in large language models, stochastic switching promotes open-endedness in sparse random Boolean networks. Another kind of clay, intelligence is shaping of intrinsic noise, and then an open discussion. So this is interesting stuff. Okay, so let's start with this example. This is Rule Hunt. This is something that was produced during one of the hackathon sessions at the Alife conference. This is a symmetric cellular automata explorer, and the idea is that you can distribute your explorations and collectively amongst players on the internet find rules for cellular automata. I guess that's what's going on here. So, we can set up a simulation, we can set up a random patch, a 10 by 10 random patch, and we can run it and see we have some patterns here being generated. These have gone extinct. We can change the number of alive cells. Let's try that. So, you have to play around with the parameters to get some interesting results. Random seed is full grid. Random patch is like that. The center seed is just where you have one cell that's active. Okay, so this is their GitHub repository. A couple of people are involved in this as contributors. And so this was just kind of put together at the hackathon. So this is a distributed exploration of the vast universe of cellular automata rules. Rule Hunt is a web-based platform for discovering interesting patterns in the combinatorial space of 2D cellular automata. They're building a TikTok-style interface where visitors can contribute their device's computing power to explore and catalog fascinating emergent behaviors hidden in the 2 to the 512 possible rules. And so, the hackathon theme for this year was exploration of emergence in complex systems, a core topic in Alife research. Well, we're not exactly on the theme. They're more about exploring the space of rules than emergence per se. This is what we were excited to build. Sometimes the best hackathon projects come from following your curiosity rather than the rubric. So this just kind of explains Conway's game of life and how that's basically what you're trying to implement in this interface. And so the idea here is that you have this vast number of possible neighborhoods. And so they're basically reducing the space with C4 symmetry. So many of the 2 to the 512 rules are fundamentally identical. They just differ by our rotation. So these 4 neighborhoods down here, as an example, should arguably produce the same result if we don't care about the relative orientation of the game grid. So these 4 neighborhoods are all equivalent mathematically. The C4 symmetry group consists of 4 rotations that are at 90 degrees. Under these transformations, the 512 neighborhoods partition into 140 equivalence classes called orbits. By requiring our rules to respect C4 symmetry, we reduce our search space to 10 to the 42nd possible symmetric rules, which is still large, but you can explore this through this distributed computing paradigm. So they just kind of go through how this works and what they want to do for the future. It's typical hackathon stuff. I think it's very interesting. So that's the kind of thing people do at Alife. They explore cellular automata, but there are many other things that people talk about there and are interested in. So that brings us to Michael Levin's keynote. So Michael Levin was one of the keynote speakers this year, and he has a post on forms of life, forms of mind, and this is his talk here, where he talks about a robot scientist, which is a translation tool between diverse intelligences. So the idea here is that there's this process of scientific discovery that adds an active inference loop into it, where you have a robotic agent on one end and a scientist on the other that can sort of supervise it, I guess. There's also kind of including agential materials into this talk. So this was, I didn't attend the talk, but, it was in that area of his, he's given a lot of talks on that. And this is, of course, in consultation or in collaboration with Josh Bongard. This is the ICDO project, which is this Institute for Computationally Designed Organisms. So this is the things that they're doing with xenobots. Okay, so this is a Alife keynote talk, the embodied mind of a new robot scientist. And then this just shows like some of the things that they're getting at with this talk. Okay, so that was interesting as well, and there are a lot of themes in his work that kind of come out in this talk. So why don't we get into some of the screenshots? Hiroki Sayama on Blue Sky was one of the main people who is putting out a lot of these screenshots on social media. So thanks to him for doing this and kind of covering the conference, covering all the different things that were going on at the conference. There were a lot of things both in the workshops and in the main sessions that were interesting. So this is another cellular automata study, Sydney Pontasfilio, using critical neural cellular automata for reservoir computing. So this was, some of that work there. This was Christopher Colon Lugo, who used 3D UNets to capture patterns in the game of life. So another cellular automata study where people are exploring these different rules and their results. This is Pietro Miotti proposing differential logic cellular automata. So there are a lot of different types of cellular automata studies. The conference was very rich in that. And it looks like what they're doing is they're doing a lot of the differential cellular automata work. I think we've talked about this work before. This is like kind of related to the neural cellular automata work, as well as the differentiable cellular automata work. And there's a 2021 paper from the Still journal, which is kind of the place you would go to look at. It looks like some of these examples were kind of taken from that paper or inspired by that paper. So that's all very interesting work. This is Aimi TK on Blue Sky, introducing us to his computational framework of bio-inspired adaptive systems using social allostatic principles. He followed up his model showing empirical testing of it. So this is work on allostasis, actually. And there were a couple pieces of work in the conference on allostasis. And so this is interesting. This is Marko Savechko exploring the space of Floblenia. So this is based on Floblenia. This is we talked about this paradigm, I think, maybe last year, sometime late last year, where it's actually the Lenia platform, and then there are different versions of Lenia. So there's regular Lenia, there's Flowlenia, there's some other versions of Lenia where people can use something it's not quite a cellular automata. It's this open artificial life platform that allows you to explore behavior and other aspects of artificial life. This person was from the Flowers AI and Cognitive Science Lab at INRIA in France. So I'm glad to see they're represented there. This is Nathaniel Virgo, and so Nathaniel Virgo, I guess. So Alife 2025 was hosted in Japan. There's a whole group of people in Japan doing Alife. One of the groups is the Cross Lab, and Nathaniel Virgo, up until recently, had been affiliated with the Cross Lab, and so he's done a lot of stuff on different types of adaptive systems. This is where he's talking about a good regulator theorem for embodied agents. So this is of particular interest to our group because we're interested in embodied agents and we're interested in a good regulator theorem. So this is some of his notes on this idea. So let's zoom in a little bit here. I think right now he is somewhere, I think at University of Hertfordshire in England. So that's where he is right now. But this is where he's kind of coming up with some ideas about using information theory and other types of tools to look at embodied agents. Kind of thinking about the Evergood regulator theorem, here's the famous diagram where you have the mapping between the model and the world, and then this domain, which tries to approximate this target domain on a domain of the world. So Z is the domain of the world, G is the domain, the target domain, the model, R is trying to approximate that from system S. And so, this is the diagram that Ashby drew out in the Connaughton-Ashby paper. And then we also have this, of course, quote, every system that is a good regulator of itself must have a model of its environment. And so this is actually, he makes the connection with the notion of meaning and enactivism. So interesting stuff there, and kind of related to, thinking about the good regulator theorem and kind of bringing it forward into modern artificial life research. Okay, this is Blaise Agaric-Iarcus, and this he's showing a live demo of his famous brainfuck experiment. So this is a famous experiment in programming languages. So brainfuck is an exotic programming language invented by Urban Mueller in 1993. It takes its name from a union of two English words, brain and fuck. It plays on the words since the language is deliberately simplistic, because the expression brainfuck evokes in slang that which puts the brain in a state of confusion by its apparent complexity. So that's kind of the background here. So we've talked about this in our Saturday morning meetings, and how that's kind of a benchmark for intelligence. There's this book, What is Intelligence? And this is Blaise here. And this is the simulation down below. Okay, so this is Complexity Kit on Blue Sky. And they also put out some posts showing some of the things going on at the conference. This is the Synthetic Biology Artificial Intelligence Workshop. Antonio Flores explains the concept of the robosphere, a complex of self-organizing multi-scale robotic ecosystems capable of autonomous adaptation and interaction with life from nano to macro. So this is a slideshow here. What is the robosphere? I'm talking about that. And, so the robosphere can be defined as the totality of integrative ecologies of human and robotic agents, interconnected and or interacting across different scales, from the nano to the macro within the planetary system. This is George Musser, also on Blue Sky, kind of talking about the Blaise's talk on brainfuck, and this is kind of getting at something that happened in the talk here. Evolution has an arrow of time towards greater complexity. It's driven neither by mutation nor by crossover / sex, but by symbiogenesis. And so this is following what Lynn Margulies was talking about when she talked about symbiosis in eukaryotic cells. And so, as sort of evidence for this, Blaise showed lots of cool core war-like competing programs. So these are the core war programs running and demonstrating this principle of symbiogenesis. This is Complexity at CAT again. How can the physics of the game of life be modified to create extended non-equilibrium conditions? What new structures could be created with these changes? Hank Rainwater addresses these questions, showing self-organization and phase transitions in driven cellular automata. So this is, again, more cellular automata work where they're showing off different, kind of basically order from randomness that you can produce through the game of life and through what they call a driven game. This is Complexity Cat once again. I start the last day of Alife 2025 at the Cellular Automata session, where Matthias Barandiran presents his work on physical reservoir computing. His approach allows us to explore new diverse structures in graph cellular automata. And so this is sort of what they're trying to produce here through reservoir computing. So reservoir computing, what they're using physical reservoir computing, is a large container of random nodes. And the idea is that through training, you can shape those nodes into a network that describes what you're trying to produce. So you're leveraging the intrinsic dynamics of a physical system, and you can use all sorts of dynamical systems as a reservoir, and then you end up producing these developmental graph cellular automata, and then you end up producing these physical structures, linear, loosely stranded, and other. This is Elias Najaro on Blue Sky. And he's, of course, known as the maintainer of the Neural Developmental Program's GitHub repo, repo that we've used in our group. So, of course, he's on the panel here of the Sony session. And basically, this is a discussion on how we can rethink intelligence under the complexity lens. Okay, this is a screenshot of Rule Hunt, which I just showed at the beginning of the presentation. This is a screenshot of flow linear. So I showed earlier flow linear. This is where you have the interface for that. This is the introduction slide for social allostasis, or how I learned to stop worrying and love the noise. This is a screenshot from the talk on differentiable cellular automata. So this was, these are the different things you can produce using logic gates. A lizard, you have 522 gates. Google logo, 927 gates. And a colored checkerboard, 465 gates. This is the 3D UNet architecture and hyperparameters. Kind of a close-up of that. This again is differentiable logic cellular automata. So they're actually using, of course, logic gates to differentiate between different parts of a pattern. So you can see that there's step N, which is kind of a weirdly misshapen lizard. And then you have 8 channels, a perception kernel, perception vector, an update circuit, and then you get the updated step, which is a more refined lizard. This is a close-up of developmental graph cellular automata. So this is where you have your initial graph, you have a neighborhood, you have this action network, you have this restructured graph, you have neighborhood information, and then you have your final graph here. So that's their kind of take on development and developmental graphs. This is, I don't remember what talk this was from, but this is where they're producing avalanches in this cellular automata. And this is evolving towards criticality, where the goal of the fitness function is to produce a power law distribution of avalanches. So the idea is you're evolving the cellular automata, and the fitness function is enforcing this power law distribution of avalanche events. And so the avalanche events basically are these cascades of activations across the cellular automata. This is physical reservoir computing again, so this is a close-up of the slide that they showed there. And this is, of course, the flow linear talk. This is where exploring flow linear universes with a curiosity-driven AI scientists, discovering diverse ecosystem dynamics. And so this is from the social allostasis talk. The question here is, how do systems maintain stability in unpredictable environments? And this can be a wide range of interacting systems, such as biological, artificial, individual, and social systems. This is, of course, going back to the 3D UNets talk. This is leveraging 3D UNets for the recognition of emergent patterns in Conway's game of life. These are the structures produced by the developmental graphs. So these are linear structures, loosely stranded structures, and other types of structures. This is, again, the reservoir computing talk. This is more flow linear. This is this book, What is Life/Intelligence? This is another screenshot from the social allostasis talk. Hormone-like signal transducers as information encoders. So you have environmental/social noise, an adaptive signal, and the resulting reconfiguration of internal states and parameters. And so this is just kind of talking about how this process works. This is the good regulator diagram here. I thought this was really interesting, sort of the way that it was defined. This is, of course, from Conant and Ashby, this figure. Their definition of good regulator involves a good set. In our case, this will be the subset of good states. So this is where you get, this definition and the good regulator theorem of something that's good, which is, of course, you could, that's shorthand for maybe something that conforms to a fitness function, or something that's in a certain range of optimality, or something that is functional versus non-functional. So there are different ways you can define good. But of course, the idea is you want to get to this G within Z. So Z is all states. G are all the target states or the good states. And so that means that you have to have a good set of solutions or a subset of solutions. So this is, we're going to be coming back to this because we're doing one of our research groups, our cybernetic research group, is doing research on the good regulator theorem, and really kind of pulling that apart and thinking about it in terms of like how it relates to intelligence and how also it relates to embodied systems, although we're not really interested in necessarily in activism and things like that. This is another screenshot from Nathaniel Furco's talk on the good regulator theorem. Our theorem could also be stated as every system that is a good regulator of itself must have a model of its environment. Again, the definition of the ever good regulator theorem is that every good regulator should be a model of a system, or like a good or optimal model of that system. So the idea is you have a model of a system. A system exists in the world, and a model is created of that system. And that model should be good in the sense that it captures all of the relevant information of the system. And so the good regulator is something that has a good model and then interacts well with its system or its environment to capture changes. And then that representation is good. And so Ashby was, I guess, interested, it's kind of the language there is a little bit vague because they're always talking about a system, but the system could be anything. It could be biological, it could be social, it could be any kind of system in the world. And the idea is that, you're talking about this very general regulation. So anything in the world will have a model, and then that model will have be able to capture some of the changes. But of course, we're talking about a wide range of different types of systems. So it's hard to really get a fix on what exactly are the details. And sometimes, social systems and biological systems have different properties where those models, how those models are updated and things like that. So if we're talking about social system, if we're talking about an intelligent agent, versus if we're talking about a biological system where there's not necessarily an intelligent agent, does updating of that model proceed in the same way? Or does that system have the capacity to build a model? We're playing around with some ideas in terms of putting an observer into this loop. And so we are actually in our arXiv preprint that we have on this, and I'll point you to this in the notes of the YouTube video. This actually has an interesting set of properties that are beyond this definition. So this is more reservoir computing. This is where we have a flattened representation plus a neural cellular automata. We have this linear SVM and then class prediction. So they're just kind of showing using reservoir computing to, testing it out with an NCA using MNIST classification. So a very simple classification benchmark using NCAs and reservoir computing together to make those predictions. These are configuration graphs. These are from a Moore neighborhood and a cellular automata just showing like how different configurations, which are living connected cells, can be represented in a graph or in a cellular automata. This is talking about criticality and how this is useful for reservoir computing. So this is a reservoir computer. This is the reservoir here. All these random nodes that get connected from input layer information. And then when they get connected, then it produces an output layer that is not as predictable as you might have if you had like a well-trained network. So this is a fixed connection, and this is a trainable connection in the dotted lines. So the fixed connections are things we already fixed from the input layer, and then the dotted lines, the trainable lines, are things that form from the input layer to the reservoir, and then of course from the reservoir to the output layer. And so their point here, I guess, is that because of the structure that you have here, you have criticality that's possible, where you can have these sudden changes that happen. Okay, so one of the reasons they're using the Everett regulator theorem in the Everett regulator theorem talk was that they want to be able to reconcile a number of traditions for looking at intelligent systems using information theory or not using information theory. So, there's the analysis of dynamic agents, there's semantic information, there's as-if contingency, the free energy principle, and integrated information theory. So a lot of things revolving around active inference and dynamical systems and different types of concepts of agency. And so they want to kind of work on kind of unifying all those approaches. But they don't necessarily know how to approach evaluating information in an agential system. So they say we want to do something similar, but we don't just want to know which information is meaningful. We want to know what it means. So they want to have some accounting of the semantics or of the meaning. And it's what in one of the previous slides, they said this is consistent with the concept of meaning from an activism. Okay, so we don't use information theory for this, although perhaps in the future we will. But instead, we present a theorem, which is the Everett regulator theorem. And then finally, this is where this talk on social allostasis, where this is from Shulkin 2011, where they make the distinction between homeostasis and allostasis. So, and then adding the word social. So this definition is the social environment contains useful information for further adaptation. And so this just shows the environment and the agent and how you get these perturbations that change the agent's internal state relative to the environment. Okay, so that was Alife 2025. It looked really interesting. Let's talk about a few other things before we go. So I was revisiting some ideas from our previous year's activities. One of these was in a talk that I found from 2016. This is following up on this work that we've been doing with like lineage trees and differentiation trees and thinking about morphogenesis. And these models that were in this talk that I was just pulling things out of was something called polygonicals, I guess would be the word. Okay, so it's this thing called polygonicals. And so this is something that we're working on way back when. And this is the idea where over time we can use these abstract shapes and we can partition these shapes. And we can then use Euler paths to analyze them. So the idea here is you have these polygons that you partition internally, and then you evaluate those partitions for viability by them following an Euler path. And an Euler path is where I can take each edge of this compound polygon and I can draw a line through it once and only once. So in other words, each line I should be able to draw, or each edge I should be able to draw a line through once without drawing a line through any edge twice. And so if I can do that, it satisfies the criterion of an Euler path. If not, that compound polygon has an incomplete Euler path and it's non-viable. It's a way to look at partitioning within embryos and partitioning within biological vesicles and cells that describe sort of its optimal shape, its optimal partition. And so this is something we've been working on this over the course of the DevoWorm program. So this was kind of something that emerged in 2016, and we've been kind of working on it ever since. And so we haven't really, I can't point you to a paper on this because never really put it into a paper. It's one of these things that's pretty far out there and it's pretty theoretical, so it's hard to really kind of get a good publication out of it. But perhaps to write something up by the end of the year for that. So finally, I wanted to talk about a range of biophysical topics. So this goes back to a set of social media posts by Ulrich Schwartz, who's a biophysicist in Germany. And so he was doing this really interesting analysis of sporozoites and how they kind of transport in an environment. So sporozoites are these malaria parasites, and they're interested in looking at their collective migration and the mechanical flexibility of the sporozoites. So Plasmodium sporozoites are the crescent-shaped forms of malaria parasites injected from the salivary glands of mosquitoes into the skins of their vertebrate hosts. So and then this is the paper that is talking about collective migration reveals mechanical flexibility of malaria parasites and method in nature physics from 2022. And so here's some example of the sporozoites, and they exist in these vortices. And so the malaria parasites form these collectives within rotating vortices. And so these sporozoites are injected by a mosquito into the skin, and they form these vortices in which they reside. And so that's the kind of collective behavior he's describing here, where he's doing agent-based modeling, or the group is doing agent-based modeling to figure out kind of what the propulsion dynamics are and what the physical attributes of that are in terms of elastic energy, and how that can be extended to understanding elastic matter. This is another paper related to this, the power of parasite collectives. Plasmodium sporozoites can move in rotating vortices owing to their chiral shape and mechanical flexibility, revealing important physical aspects of collective motion. So we can see that these vortices serve as sort of these organizing structures. If you look at fish, they often form vortices as sort of these collective behavioral structures. And they're very similar to these vortexes of sporozoites. These vortices are self-sorting, which means that there's sorting that happens within the vortex based on speed and curvature. And then these vortices don't just pop up. They're self-organized, but their self-organization are based on a number of key ingredients. So first you need chiral movement, which is movement along a curve, and it kind of translates into this circular motion. So you need this sort of chiral orientation. In this case, it's kind of bending to the left, I guess. So it might be considered left-handed. And then you have these steric interactions where you have movement of these chiral vectors and kind of moving them close to one another or apart from one another and finding the optimal distance between one another. So you can see that these chiral movements, actually each of the parasites have their own chiral movement in this vortex. And then these chiral movements are separated by a certain distance. Sometimes they're nearly overlapping, other times they have a little bit more distance. But they try to maintain sort of a healthy distance without being too far apart. And then finally, mechanical flexibility. So sometimes these vortices can maintain their integrity well under conditions of turbulence. So you get this bending of the chiral movements of the form, and you end up with this stable vortex. And so they kind of described all of this and the mechanical flexibility here. And that's just another paper kind of highlighting this. So this is another social media post by Ulrich Schwartz. The bridge between experiments and models is quantitative image processing. This revealed that sporozoites in the vortices are sorted according to speed and curvature that the vortices undergo shape oscillations. So again, there's this adaptive feature. So this is all from the May 30th meeting in 2022. And so it's interesting to kind of revisit that. But I wanted to go back to some of that work. This is a new paper that the group has put out. This is three-dimensional chiral active Ornstein-Uhlenbeck model for helical motion of microorganisms. And so this is a brand new paper. It's on the arXiv, and it describes active movement being essential for the survival of microorganisms like bacteria, algae, and unicellular parasites. In three dimensions, both swimming and gliding microorganisms often exhibit helical trajectories. So this is where you have microorganisms that move around their environment, their liquid environment, and they exhibit these helical trajectories. Oftentimes you'll see like they might have a rotor on their flagellum, or they might just move helically, which means they move in these circular motions outward. So they're moving in this helix. They produce this helix as a sort of a trace of where they've been. One such case are malaria parasites gliding through 3D hydrogels, for which we find that the internal correlation time for the stochastic process generating propulsion is similar to the time taken for one helical turn. So this is where, there's this stochastic process that produces the propulsion. You get this helical turn, and there's this correlation between them. Motivated by this experimental finding, here we theoretically analyze the case of finite internal correlation times for microorganisms with helical trajectories as chiral active particles with an Ornstein-Uhlenbeck process for torque. So describing the torque component of those movements. We present an analytical solution which is in very good agreement with computer simulations. We then show that for this type of internal noise, chirality and rotation increase the persistence of motion and results in helical trajectories that have a larger long-time mean square displacement than straight trajectories. And this is at the same propulsion speed. Finally, we provide experimental evidence for this prediction for the case of the malaria parasites. So this kind of talks about, I know this is from the stuff that I talked about from 2022 and kind of applying it forward to this paper today. However, most of these studies consider 2D cases, whereas helical motion of chiral active particles occurs in 3D. So they're extending this to the third dimension or the 3D case. If chiral active motion in 3D was analyzed theoretically, but mostly in the context of swimming, an early work on asymmetric swimmers in 3D considered the analogy to polymer models to extract new power laws for effective diffusion. In general, the statistics of fluctuating helices is also an important aspect of helical biopolymers like DNA. Previous work on sperm swimming considered stochasticity on the level of curvature and torsion and showed that helical trajectories are useful search strategies for chemotaxis in noisy environments. So this is all to say that, this isn't just studying some, random phenomena. This has implications for chemotaxis, for chemotactic following, for motility, and then, of course, how microorganisms interact with their liquid environment. So this is kind of getting at a lot of these different issues all at once. This line of work also considered colored noise in the form of a power spectrum. One study of chiral motion in 3D started from the full mobility tensor for an arbitrarily shaped particle, and showed that helical trajectories are the most likely outcome. Similarly, a chiral active Brownian particle model has been used to describe the helical motion of planoflagellates and to show that purely stochastic propulsion can result in effective dispersion. Very recently, it has been shown in a deterministic model for sperm swimming that an asymmetric beat of the flagellum leads to helical trajectories with high persistence. Collectively, this body of work demonstrates that helical trajectories can have evolutionary advantages for microorganisms. So this has to do with noise and how this relates to sort of how noise is used in motility, how noise is produced by biological systems. And sometimes noise is a good thing as well as a bad thing. One of the sessions at the Alife conference talked about noise in biology. And, noise, we often think of noise as deleterious or bad, but sometimes noise can drive different processes and augment them and improve them in different ways. Sometimes we fight noise, and in fighting noise, we actually have these sort of adaptive evolutionary traits that arise. So we can do things to buffer or counter noise. And so this is how they're modeling this. You look at an Ornstein-Uhlenbeck process, and this usually replaces the body fixed constant velocity with a noisy velocity performing an OU process around a body fixed average. So they're modeling these movements with noise, and they're able to sort of understand these things in 3 dimensions. And so this is kind of showing these reconstructed trajectories of the malaria parasites producing these chiral motions outwards of these spiral vectors. And this is where they're gliding through a synthetic hydrogel. Because this environment is nearly isotropic, meaning there's very little heterogeneity in the environment, the right-handed helical trajectories persist for a long time. So you can see that they can produce these very long trajectories that persist, that don't have to shift their motion in any one direction. They can produce these very clear signatures of this spiral movement. Okay, so that was that paper. And I wanted to get to this because our group was interested in this topic, but Ulrich Schwartz in his lab, they've also produced some work on syncytial embryos. And so this is from the Journal of Theoretical Biology. This is from 2014, and this paper is a computational model of nuclear self-organization in syncytial embryos. So this is where they present an individual-based simulation of the Drosophila melanogaster embryo, focusing on dynamic changes in internuclear forces throughout the cell cycle. Repulsive forces are necessary and sufficient to effect correct nuclear self-organization. Cytoskeletal networks must reform quickly after mitosis, and disordering is nonlinearly correlated with spindle elongation. So if you remember, we talked about syncytial structures coming from cells that fuse together, and as the cells fuse together, the nuclei can divide. So you can have nuclear division. So you can have 2 cells that merge together. That new cell will have 2 nuclei. And then those nuclei can divide, so your syncytial structure can consist of 2 cells but have many more nuclei. And so all these things require different types of internal forces in the cell. You need to have modified cytoskeletal networks as cells fuse together, and you get different structures within the cell. And so you go through this set of nuclear divisions and a set of cell divisions. And so this is an interesting system because we generally think of cell divisions as including the nucleus and the cytoplasm and everything that kind of segregate out and divide. And that's not what we see with syncytial structures. We see sort of the opposite, or we see very complex nuclear dynamics. So this is, let's go through the abstract. Syncytial embryos develop through cycles of nuclear division and rearrangement within a common cytoplasm. A paradigmatic example is the Drosophila melanogaster, in which nuclei form from an ordered array in the embryo surface over cell cycles 10 through 13. So Drosophila melanogaster is a famous example of how you can have these syncytial structures at a very large scale. And so this is the example they give. This ordering process is assumed to be essential for subsequent cellularization. So you have the syncytial structure that breaks down into different cells. So in this case, you get this syncytial structure that builds up. You get a single cell that has a large number of nuclei. And then each of those nuclei, then eventually you get these different cells that cellularize. And then you proceed with development. So, we have these different dynamics. We have cell fusion, we have nuclear division, then we have not cell division necessarily, but cell differentiation, and what they call cellularization. So that's what they have here. Using quantitative tissue analysis, it has been previously shown that the regrowth of actin and microtubule networks after nuclear division generates reordering forces that counteract its disordering effect. We present here an individual-based computer simulation modeling the nuclear dynamics. In contrast to similar modeling approaches, such as epithelial monolayers or tumor spheroids, we focus not on the spatial dependence, but rather on the time dependence of the interaction laws. We show that appropriate phenomenological internuclear force laws reproduce experimentally observed dynamics, provided that the cytoskeletal network regrows sufficiently quickly after mitosis. So you have, all these different things going on. Then repulsive forces provided by the actin system are necessary and sufficient to regain the observed level of order in the system after the strong disruption resulting from cytoskeletal network disassembly and spindle formation. So you still have spindle formation and you have changes in the cytoskeletal network that need to be, they need to be overcome. So this is, these are all things that we can model. We also observe little mixing of nuclei through cell cycles. Our study highlights the importance of the dynamics of cytoskeletal forces during this critical phase of syncytial development and emphasizes the need for real-time experimental data at high temporal resolution. So this is just showing the system in Drosophila, where they have these different nuclei. You have this mitotic wave, as you would have with individual cells, and you get these nuclei that undergo mitosis. They divide, and then they form these cells that are lined up. And then in cellularization, each of these nuclei have a cell that forms around it. So this is where they kind of show the dynamics here of nuclear division. You have this mitotic wave, you have nuclear division, you have interphase, and then mitosis, which show the spindle formation and spindle dynamics. And so there are these interphase force laws, which we can use to describe interphase as a step in nuclear division. This is their simulation where they show the syncytial embryo. Starting from interphase, the embryo undergoes a period of mitosis, with nuclear division being initiated at the poles of the embryo. So in these areas up here and down here. Subsequent to mitosis, daughter nuclei are driven apart by the mitotic spindle. So you have these, the poles are in red, the sort of the midsection is in blue, and then we have nuclei that are driven apart by the mitotic spindle. So you see here, and then before reordering is C, and then D is nuclear color indicates cell age. So this shows that. Okay, that was a good paper. So yeah, and then finally I wanted to highlight Ulrich Schwartz. He's at Heidelberg University in Germany, and he has a number of publications here that you might want to look into. He's done a lot of things in biophysics, really interesting modeling and physics, looking at theoretical soft matter and biological physics. All right, so that's all for today. Thanks for watching, and see you next week.